Alright, mic check, 1, 2. Let's get ready to science, guys. Let's get ready to science. I turned the freeze off on the board. Yes, I did. Alright, I am going to apologize now for sitting. I am not my best today, and I'm going to attempt to sit while I do this. Not the best at sitting down while I teach. I just can't. Feel like I'm not as effective when I do that. But, whoa. See what I'm saying? Even the mic don't want to act right. Alright, guys. Welcome back. This is the last day of the week that you will get to see me. When I see you again on Monday of next week, you will have a quiz on the major organic molecules and the introduction to organic chemistry. So you will have a quiz on Monday. Go ahead and take out your note packets and close your MacBooks for me, please. If you are in the middle of completing that review, it is timed, so you need to complete that and then close your MacBook, okay? But if you have already completed your review from lesson 2, then you can go ahead and close your MacBook as we begin lesson 3. In your note packets, you need to be on page 78, I think. Alright. Awesome sauce. Needs to be on page 78. Quick review of what we've discussed thus far. We talked about organic chemistry being based around the element carbon. And carbon makes 4 covalent bonds so it can form a bunch of diverse structures. We care about that because biomolecules are those molecules synthesized by living organisms that they need in order to survive. In particular, we care about the 4 major biomolecules or major organic compounds known as macro, because macro means big, macromolecules. Those macromolecules are carbohydrates, lipids, proteins, and nucleic acids. Last time we discussed carbohydrates and lipids. We talked about the fact that carbohydrates serve as a source of short-term energy storage, and they're called carbohydrates because they are composed of carbon, hydrogen, and oxygen only. We talked about the monomers being called monosaccharides, mono meaning 1, saccharide for sugar, and that the polymers can be disaccharides, 2 sugars, or polysaccharides. We talked about the fact that the energy in these major organic compounds is located in the bonds between carbon and hydrogen. The energy is in the bonds between the carbon and the hydrogen. We talked about the different polysaccharides, the classes of polysaccharides that we can find in plants and animals, saying that some are used for structure, so it helps to build up that plant or animal, and others are used for storage of excess glucose molecules, and we know glucose is our most common monosaccharide. We talked about the animal storage molecule being glycogen, and the plant storage molecule being starch. The animal structure molecule, which is found in only some types of animals like arthropods and insects, is chitin. Chitin is also found in the structure of fungus cell walls. And we talked about the plant storage molecule being cellulose, which is found in the cell walls of plants. After we finished carbohydrates, we moved on to lipids, and we compared the energy you can get from a carb to the energy from a lipid. We talked about how carbohydrates only give you 4 calories per gram, but carbohydrates give you 9 calories per gram. There are 4 classes of lipids, and those lipids are triglycerides, phospholipids, steroids, and waxes, with the triglyceride being commonly known as fats and oils and being used for long-term energy storage, insulation, and protection of internal organs. The phospholipid contains phosphorus, and in addition to the carbon, hydrogen, and oxygen, and it is used to create the plasma membrane or the cell membrane. We talked about steroids being used as chemical messengers, and that the most common one is cholesterol, and is used to help build those sex hormones. And we talked about waxes being used for protective covering. Now, all of our major organic compounds contain carbon, hydrogen, and oxygen, okay? All 4 of them contain those 3 elements. So now we're going to move on to the nucleic acid. The nucleic acid is extremely important because the nucleic acid is the one that is responsible for making our DNA, and our DNA is the hereditary molecule. So the nucleic acid is composed of the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. Don't forget, you should be filling out that table that you got in class last time as we go through these, as well as any missing information you may have in your note packet. So you want to have both of those things available as we go through this, okay? All right, so again, the elements that you would find in your nucleic acids are carbon, hydrogen, oxygen, nitrogen, and phosphorus, okay? Now, the nucleic acid is the only one that will have all 5 of these elements present every single time. The monomers for a nucleic acid is called a nucleotide, or some people pronounce it nucleotide. So that's the monomer for the nucleic acid, okay? Now, what is a monomer, by the way? The what? Well, the monomer in a carbohydrate is a simple sugar, but monomer is a generic term for the building blocks, okay? So when you think of monomer, you can't only think of the simple sugar. If you're thinking simple sugar, that's just carbohydrate. But the other major organic compounds also have monomers. And in this case, the monomer for nucleic acid is the nucleotide. Now, nucleotide has 3 parts of its own. It is made up of a sugar, a phosphate, and a nitrogen base. So let's look at the structure. If you didn't get it, you can write it on the next page. Let's look at the structure of a nucleotide, and I want you to take time and draw it in that box on page 78. Take time and draw this nucleotide and label it in the box on page 78. As you're drawing it out, now you don't have to try to put all the elements, you can just draw the shapes. Like for phosphate, you can put a circle. For the sugar, you can make it kind of like a house, and for the base, you can do like a double ring or a rectangle. What you want to do is just make sure you know the different parts. You don't have to try to draw the actual chemical structure because it is already there for you on that page, okay? All right, so a nucleotide, which is the monomer for a nucleic acid, is composed of 3 parts, and you do need to know these 3 parts and where they're located. There is a phosphate group that will be attached to this carbon. This is the number 5 carbon. There's a phosphate group attached to the number 5 carbon. On the number 1 carbon, there's a nitrogen base. Okay? So what I'm saying is on one end, there's a phosphate group. On one side of the sugar, there's a phosphate group. And on the other side of the sugar, there's a nitrogen base. Now this image can flip. Okay? So don't say, well, it's phosphate on the left and nitrogen on the right. It can be drawn when nitrogen is on the left and phosphate is on the right. What matters is that you know that the phosphate is on one side and the nitrogen is on the other side. The nitrogen base is on the other side. And in the middle, we have a 5-carbon sugar. Does anybody know the prefix for 5? Penta, right? So, and what do most sugars end in? Ose. So this is called a pentose, okay? This is a 5-carbon sugar called a pentose. Pent for 5, ose for sugar. So the sugar is a pentose. There's a phosphate group, and there's a nitrogenous base. That's the basic structure of a nucleotide. Now there are 5 different nucleotides. There's 5 different nucleotides, and they differ because of the sugar and the base. Okay? Well, technically there's more than 5 because if you change the sugar. But anyway, we're going to stick with 5, okay? All right. Now, the sugar, which is right here, can be one of 2 different sugars. It can be a ribose or it can be a deoxyribose. If you look over there on the sugar, you see ribose and deoxyribose. What do you think deoxyribose means? Not no oxygen, but less oxygen. That was a good guess. It means less oxygen. So the sugar deoxyribose has 1 less oxygen than ribose. So ribose has 3 oxygens and deoxyribose only has 2. So this sugar right here would be a deoxyribose because there should be another oxygen right here, okay? If a compound contains a deoxyribose, if that nucleotide has a deoxyribose as the sugar, then you should know that that is DNA because the D in DNA is for the sugar, which is deoxyribose. DNA's name is deoxyribonucleic acid. So it tells you what it is. If it's deoxyribonucleic acid, then you know it has to be a nucleic acid. Which sugar is present in it? Deoxyribose. RNA stands for ribonucleic acid. What type of sugar is in RNA? Can't hear you. Ribose. Right. Because most sugars end in ose. So the sugar in RNA would be ribose. Another place where these nucleotides can be different is right here at the nitrogenous base or the nitrogen base. You don't have to write nitrogenous, you can write nitrogen, okay? Because look at this base. What's most of the elements in this one that you see? Nitrogen. Now, of course, we know that we have a carbon here, 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 and here. But what we see mostly would be the nitrogens, okay? This is a nitrogen base. Now, I want you to notice something about this base. How many rings do you see here?
2.
2. Some of the nitrogen bases have 2 rings. In fact, 2 of the 5 have 2 rings. The other 3 only have 1 ring, okay? So, the 2 bases, adenine and guanine, have 2 rings. And because they have 2 rings, we call them purines. P-U-R-I-N-E-S. Purine equals 2 rings. Okay? Make a note of that. Purine equals 2 rings. And there's only 2 bases with 2 rings. Can you put 2 and 2 together? The 2 bases are adenine and guanine. You can put A and G. You don't have to, as long as you know what the A and G stands for. You have to know that A is for adenine and G is for guanine. Now, the other 3 bases are what we call pyrimidines. Pyrimidines only have 1 ring. Okay? And the 3 bases that are pyrimidines are cytosine, uracil, and thymine. You want to know how I remember that? I say cut pyrimidines. Because the word is big and I want to cut it down. I get tired of saying it. Cut pyrimidines. But if you can remember to cut pyrimidines, then you'll remember that the three bases that are pyrimidines are cytosine, which starts with a C, uracil, which starts with a U, and thymine, which starts with a T. So what do we want to do to the pyrimidines? What does CUT stand for? Cytosine, uracil, and thymine. So 3 bases are pyrimidines, and the other 2 bases, which are adenine and guanine, are purines, okay? That's important because when bases pair, when we talk about it later on, a purine bonds to a pyrimidine. But we'll talk about that in another unit. But let's look at, actually, I'm talking about it right now. My bad. Let's look at an example of a polynucleotide, okay? The monomer for a nucleic acid is called a nucleotide, and the polymer is called a polynucleotide. Easy enough to remember, right? What's the monomer? Can't hear you. Y'all usually more responsive than these. Come on, talk to me. I don't feel good today. You gotta help me out. Nucleotide. And what's the polymer? Polynucleotide. We're gonna do it again because I can't hear you. What's the monomer? And what is the polymer? Awesome. DNA and RNA are examples of polynucleotides, okay? They are examples of a polynucleotide. Now, if I look at this example of a polynucleotide, how many strands do I see here? 1 or 2? There's 2 strands, okay? Keep that in mind. I also have to be able to pick out a nucleotide. So this represents my base, because I know one of my bases is a G, so this is guanine. What does this S represent? Represents the what? The sugar. I can't hear y'all. Don't maybe make y'all stand up and do a jumping jack or something to get that energy flowing. All right, so what does this S represent?
Sugar.
The sugar. And what's the P?
Protein.
Not protein. What's the other part of a nucleotide? The phosphate. So say base. Say base.
Base.
Sugar.
Sugar.
Phosphate.
Phosphate.
Do it again.
Base. Sugar. Phosphate.
Do it with me. Base, sugar, phosphate. All right. And these 3 parts together make up what we call a nucleotide. Now, what do you notice about the phosphate on one base and the sugar on the next base? I mean, the phosphate in one nucleotide and the sugar on the next nucleotide. What do you notice about it? What do they do?
They all have phosphate and sugar.
They do what?
They repeat.
Okay, they're repeating. Okay, look at it this way. If this represents 1, from here to here is 1 complete nucleotide, then what is this phosphate doing with the next complete nucleotide? It's what?
Sharing.
It's bonding. It's a covalent bond. They're sharing electrons. You're right. So the phosphate in one nucleotide bonds to the sugar in a next nucleotide. This is called the backbone. The backbone of nucleic acids is a repeating sugar, phosphate, sugar, phosphate, sugar, phosphate. So we say that nucleic acids have a sugar phosphate backbone. Okay? Are y'all with me? What's the backbone made of? Okay. So the phosphate of one nucleotide bonds to the sugar in the next nucleotide. Now what are these things right here? Bases. What do you notice that's happening in the base in one strand with the base in the next strand? They're bonding, okay? Anytime you see lines between something, that indicates a bond. Are y'all with me? Now, this is a dotted line because this kind of bond is more of an attraction. The bond you see right here with the dotted lines, write this down, it's called a hydrogen bond, okay? The bases bond together using hydrogen bonds. You see how the backbone is a solid line? Y'all see that? This is because this is a covalent bond. Okay? This is a strong covalent bond. But this right here is a hydrogen bond because we don't want that bond to be permanent. We need that bond to be able to break and reform. Okay? Y'all with me? All right. Now, base pairing is specific. Base pairing is specific. Adenine is a player. Adenine hangs out in DNA, and it bonds with thymine. But when adenine goes to hang out with RNA, it bonds with uracil. Guanine and cytosine are exclusive. In DNA and in RNA, guanine will only bond with cytosine. Cytosine will only bond with guanine. Okay? They are exclusive to each other. But adenine, because uracil is not at DNA, it bonds with thymine. When adenine is in RNA, because there's no thymine, it bonds with uracil. So if you see thymine in a nucleic acid, you should already know that it's DNA, because there is no thymine in RNA and there's no uracil in DNA. Did you get that? Can I change one of these bases to a U?
No.
Why? Because this is DNA. Cool beans. And it wouldn't bond with cytosine anyway. Not correctly. Alright, so again, the polymers are called polynucleotides. The backbone is a sugar phosphate backbone. And the bases bond with hydrogen bonds. Couple of differences between DNA and RNA. What does DNA stand for in the first place? Deoxyribonucleic acid. Look right here on the cabinet. You see it with that big DNA poster? That's how you spell it. Deoxyribonucleic acid. What does the deoxy part, the deoxyribo part represent? The what? The sugar. All right. Now, so one of the differences between DNA and RNA is the sugar. Deoxyribo in DNA, ribose in RNA. Another difference is the base. We talked about that, right? AT, GC, and U. Which one of those is not found in DNA? If I said AT, GC, U, which one is not in DNA? U. Which one is not in RNA? T. Okay, cool bean. Y'all got it. Another difference is DNA is double-stranded, and RNA is usually single-stranded. You can have some double-stranded RNA, but usually RNA is going to be single-stranded. So if I see two strands, I see thymine, I see deoxyribose, that's DNA. Now, the function of DNA, make sure you write this on your sheet. The function of DNA is to store and transmit genetic information. The function of DNA is to store and transmit genetic information. Your genes, the reason why you look the way you look, the reason why your body responds to certain foods and chemicals the way they do is because of the genetic code that is encoded in your DNA on your chromosomes in all of your cells.
Can you repeat it again?
It's right here. Store and transmit genetic information. To store and transmit genetic information. Now, DNA is isolated to the nucleus, okay? Unless the cell is dividing, DNA is confined inside of the nucleus in a safe place and it cannot leave. So how can it control the rest of the cell if it cannot leave? It does so by sending its messenger, which is RNA. RNA is going to direct the creation of proteins. RNA is like a template. DNA writes the template, RNA translates it so that the ribosome can put together the protein. So the job of RNA is to create proteins. And there's three basic types: mRNA, rRNA, and tRNA. Okay? That's not the only types, but those are the types that you need to know about. mRNA, rRNA, and tRNA. And we'll get more into what that means in a later unit. So here's a little overview comparison between DNA and RNA. Which strands? How many strands do you see with the DNA? What about the RNA? Alright, so we see these 3 nitrogen bases are found in both, right? What about this one? RNA only. What about the other one? DNA only, okay? So they both have these 3, so there's 5 bases. 3 of them found in DNA and RNA. There's 1 unique to RNA. There's 1 unique to DNA. One way you can remember the one that is only found in RNA, it's the only one that has a letter R in it. Uracil is the only one that has a little R. So that should be like a trigger to say, oh, if I see this R, it has to be the unique one for yours, for DNA, RNA. Ugh. Yeah, forgive my screw-ups today. All right, so let's check and make sure you have everything you should have in your chart, and then we're going to go over these questions using the whiteboard. So, what elements can I find in a nucleic acid?
Carbon, hydrogen, oxygen, nitrogen, and
All right, carbon, hydrogen, oxygen, nitrogen, and phosphorus, or what I like to call chum. Okay? All right, what is the monomer? What's the monomer for a nucleic acid? Nucleotides. What are the 3 parts of a nucleotide? A phosphate, a sugar, and a nitrogen base, right, or a nitrogenous base. All right, what's the polymer called? Polynucleotide. Can you give me 2 examples? DNA and RNA. Right, I asked for 2 because it's the easiest 2, right? All right. What's the function of DNA?
to store and transmit information.
What's the function of RNA? All right. And on that picture, at the bottom of the handout, which one of those represents a nucleotide monomer? Which one? I hear B, I hear A. I don't understand what none of that stuff means. It's Bravo. Awesome. I don't understand the other stuff y'all was saying. Miss Young, you so extra. You knew what I said. You heard me 4 times. You looking right in my mouth. I know what you're saying right now. All right. Any questions about nucleic acids? Cool beans, get your boards out. Get your boards out. Get your boards out. Boards out. Page 79. I knew we didn't walk back there without our notes, did we? Oh, you gonna get your board? I'm sorry. You looked at me like, Miss Young, I got my notes. Don't play with me. It's a paper towel back there. It's a whole stack of paper towel back there. All right, guys and girls and guys, once again, this was your homework, so there's no need for me to wait for you to answer these questions because I know you diligently answered these questions over the last 2 days. So let's get going. In addition to carbon, which 4 elements are most commonly found in nucleic acids? So go ahead and write down those answers for me. And then when I count to 3, I'm going to look up and I'm going to share my answers with you. And you're going to compare your answers to mine to make sure I know what I'm talking about, because I might not get this right. What y'all think? I think I'm going to get it right? Sure? You got that much faith in me? I appreciate it. Yeah. Marking all right? Check the table behind you. All right, and throw it over in the trash for me. 1, 2, 3. I cheated. I did. I watched you write everything. Charlie, Echo, Golf, and Hotel are the correct answers. Again, please be mindful of those multiple answer questions, because if you get 3 out of 4 on a district test or a state test, you get 0 out of 4 points. All right? I know, right? But we want to be prepared. We want to be prepared. All right, question 2. Which 3 components are found in a nucleotide? Which 3 components are found in a nucleotide? Y'all, I'm going to pronounce that nucleotide, as long as y'all know what I'm talking about, even when I'm tired, we're going to be all right. All right, so, number 2. 1.
Hold on.
He called me short. Hey, I'm 6 feet tall. He called me short. All right, 2. 3. Cool beans. I love it when a plan comes together. Bravo, Charlie, and Foxtrot are the correct answers. You guys are killing it as usual, making me so proud. I do appreciate you working with me on my off day. All right, question 3. What is the main function of RNA in a cell? What is the main function of RNA in a cell? Main function of RNA. 1, 2, 3. Woo! Alpha all around. I love it. You guys are killing it. I am so proud. I might not give y'all a quiz. I'm lying. I'm still gonna quiz you. All right, so I ain't let that sink in. I'm gonna make you get happy. All right, 4. Which 3 of the following characteristics are the same in both DNA and RNA? Which 3 of the following are the same in DNA and RNA? So you should have 1, 2, 3 answers on your board. All right, so let's get ready to science. 1, 2, 3. All right, guys, the correct answer is Charlie, Echo, and Foxtrot. Remember, deoxyribose is only found in DNA. The sugar in RNA is ribose. So alpha was not a correct answer choice. Deoxyribose is exclusive to DNA. All right, moving right along. Let's answer question 5. All right, which of the following best represents the function of DNA in a cell? Which of the following best represents the function of DNA in a cell? I see people ready for me to see what they know, so let me see what you know when I count to 3. 1 and 2 and 2.5 and 2 3/4 and 2 3. All right, the correct answer is bravo. I love it, guys. I love it. I swear you guys are making me so proud. So proud.
Make us proud and don't give us the quiz.
I ain't that proud. Not yet. Woo. All right. Now, so our next topic of the day, which will start on the next page, is proteins. Now, let me go ahead and warn you, proteins is the most diverse major organic compound, and it takes a while to actually go through everything with proteins. So today is just an introduction to proteins, okay? I don't want to overwhelm you with too much information. Page 80. We're on page 80. All right, so our next major organic compound, the last one in our series of the 4, is proteins. Do not forget, your project is a 4-room project because you have carbs, lipids, proteins, and nucleic acids to put in your room. So now you'll have the information to start that project, which is due exactly 14 days from today on October 15th. You have 14 days. All right, so proteins. Proteins are composed of carbon, hydrogen, oxygen, and nitrogen, okay? Carbon, hydrogen, oxygen, and nitrogen. That's what makes up a protein.
I thought you said nitrogen is gonna be on your own.
No, I said nucleic acid is the only one that has all five: carbon, hydrogen, oxygen, nitrogen, and phosphorus. That's a good question. Make sure you clarify. All right. So, proteins have carbon, hydrogen, oxygen, and nitrogen. Now, they are the most diverse, and they have a variety of functions. In fact, I can think of 8 off the top of my head, but there's 3 that I want you to write down. I want you to write down structure, catalyst, and immunity. I want you to write down those 3 because they function in your immune system. Structure, catalyst, C-A-T-A-L-Y-S-T. Y-S-T. Cat-a-list with a Y. Catalyst. And immunity. We're going to go in details about the catalyst because an example would be enzymes. And we talk a lot about enzymes in this unit. All right. Now, the monomer for a protein is an amino acid. Say proteins are amino acids.
Proteins are amino acids.
Say it again like you mean it. Proteins are amino acids. Wait. Say it. Okay. There's 20 different amino acids. And those amino acids can be put together in different orders for different proteins. Okay? So there's 20 different amino acids, but the dry mass in your cells, like 60 to 70% of the dry stuff inside your cells is proteins. Okay? So proteins are the most diverse. And they join together by what's called a peptide bond. You need to know that. The bonds that hold amino acids together when they join through dehydration synthesis is a peptide bond. Guys, do not forget, whenever we talk about monomers coming together to make these big polymers, that's a dehydration synthesis. Anytime we talk about something breaking down, that's a hydrolysis, okay? So that stuff we learned at the beginning carries through. Don't forget about it. Now, this is an example of an amino acid. An amino acid has a backbone structure, which is what you see at the top, and it has a variable side chain, okay? The backbone structure will always have an amino group, which is the N with the nitrogen, so that's the NH2. It will always have an amino group, and it will always have a carboxyl group on the other end. So there's always an amino, there's always a carboxyl. Where we get the 20 different ones is right here at this R group. Anytime you see the letter R in a compound, that means that's a variable location. That means that's the spot where it can be something different there. It could be a H here. It could be a CH3 here. It can be a CH2, CH3 here. It's 20 different things that can be here. 20 different combination of elements that can be here. That's the only place where the amino acids are different, and that's what makes each one special. What's different about them, okay? Just like y'all, what's special about you is what's different. Y'all supposed to say, "Oh, Miss Young, that's sweet." No, y'all wasn't. Okay. All right, moving on. What type of reaction joins together two amino acids? Dehydration, good job. And what type of bond is formed when two amino acids join together? A peptide bond. All right, so here's the formation of a peptide bond we see here. So we have an amino acid there. We have an amino acid here. These two amino acids join together. And what molecule is created during the dehydration synthesis? What molecule is created? What do you see right here? Water. Okay? A dehydration synthesis removes water from the reactants, removes the elements from the reactants to make water. Okay? And the bond between this carbon and this nitrogen is what we call a peptide bond. Y'all with me? All right. The polymer for a protein is called a polypeptide. Why do you think it's called a polypeptide? More than one what though?
Peptide.
Peptide bonds. Cover your nose, mouth. Cover your nose, mouth. Come on, guys. We gotta stay in compliance. All right. There's more than one peptide bonds. There's many peptide bonds. If I have 103 amino acids, I got 102 peptide bonds. Okay? So it's called a polypeptide. These are some of the functions of different proteins. This is in Canvas. You don't have to write this down, but I do want you to read over it. Familiarize yourself with it, okay? Y'all hear me? All right. Now, proteins have 4 levels of structure. All right? You have a primary, secondary, tertiary, and some proteins have a quaternary. So that's just a fancy way of saying 1, 2, 3, and 4. So let me give you a little demonstration of what that is. And don't y'all make fun of me either. All right, so the primary would be just a straight chain. So pretend like there's a bunch of amino acids on this paper and it made a straight chain. Y'all with me? That's primary. It's just a chain of amino acids with the peptide bonds. Now a secondary would be that chain could do one of 2 things. It can fold into a beta sheet, or it can twist into an alpha helix. So I can't twist this paper, so I folded it into a beta sheet. That's called a beta pleated sheet. So that's the second thing it does. So first thing, it forms a straight chain. Then the chain folds or twists. And then after it folds or twists, it folds some more into its final conformation called a subunit. Okay? That's the tertiary. Now, some proteins have multiple subunits. So this one wouldn't fold anymore, but if this was a subunit, it might bond with this one. And then you have what's called quaternary. So let's look back at it. Primary, straight chain. Secondary, either pleated sheet or twisted alpha helix. Tertiary, folds into its final conformation. Okay? You don't have to have quaternary, but if I'm having a quaternary structure, then this one might bond with another one. It could be the same kind, or it can be a different kind. Are y'all with me? 4 levels. So this is what it kind of looks like. All right, primary. This right here, this is the alpha helix right there. I folded the paper for a beta sheet. That's the alpha helix. Tertiary structure, it folds into its final conformation. Why is that important? Say structure dictates function.
Structure dictates function.
Say it. I love the way you said that, Muhammad. Y'all say it again, just like Muhammad said it.
Structure dictates function.
All right. So what happens if I change the structure of something? It changes the function. If you still have the kind of car you got to stick a key in to crank it up, if I break your key, can you crank it up? Negative. All right. So if I change the structure, you change the function. Same thing with proteins. And that's important that we know that, because proteins are specific for pH, temperature, and salinity. Salinity means the amount of salt that's actually dissolved in aqueous solution. If I change the pH too much, my protein begins to unfold. If it begins to unfold or denature, then what has happened to it? Will it continue to work properly? No, because when you change the structure, you change because structure dictates, and what determines the function? And what the structure dictate? Change the structure. Good job, change the function. All right, so we're almost done. Repeat after me. Carbs are made of glucose.
Carbs are made of glucose.
Energy is so sweet.
Energy is so sweet.
All right, so that first line tells me that carbohydrates are made of glucose. And in case I don't know what glucose is, it tells me energy is so sweet. So when I think of sweet, I should think of sugar. And what do I get from it? Energy. That first line gives me structure and function of carbohydrates. The second one talks about the most common lipid. So lipids or triglyceride, fatty acid chains in your seat. All right, so say that. Lipids or triglyceride,
Lipids or triglycerides.
fatty acid chains in your seat.
Fatty acid chains.
All right, what's a part of the triglyceride? It has 3 what? Fatty acid chains. All right, now proteins are amino acids.
Proteins are amino acids.
Peptides make you buff.
Peptides make you buff.
All right, so proteins, what's the monomer for protein?
Amino acids.
Amino acids. And what are the peptides? The bonds, so they make you buff because protein deals with structures or muscles, which I have none of right now. All right, nucleic acids, nucleotides,
Nucleic acids, nucleotides.
keeps all your genetic stuff. Where are your genes? And your DNA. And DNA is a type of what? Nucleic acid. So this is how this going. Y'all got to do it with me at least 2 times or we're going to be sitting in here past 1:15. Y'all going to be hungry and I'm going to be hangry. Y'all know what hangry is? Okay, don't make me hangry. You won't like me when I'm hangry. Alright, so it goes like this. Carbs are made of glucose. Energy is so sweet. Lipids or triglyceride, fatty acid chains in your seat. Proteins are amino acids. Peptides make you buff. Nucleic acids, nucleotides keep all your genetic stuff. Y'all with me? It works. I promise you. Look, you'll be hearing, if you practice this or listen to it, you'll be hearing it on the test in your head. And it will help you pass the test. I promise you. I know it's corny, but it works. Okay?
You should make that a song.
I have a YouTube. It's called Young Science Rocks, and it has all my science content songs on there. So you're welcome to go to it. 1, 2, ready, go. Carbs are made of glucose. Energy is so sweet. Lipids or triglyceride, fatty acid chains in your seat. Proteins are amino acids. Peptides make you buff. Nucleic acids, nucleotides keep all your genetic stuff. I will come around and make you do it one by one, except for the people that are here. If y'all don't get it with me, get it with me. Come on, one by one. Come on. 1, 2, ready, go. Carbs are made of glucose. Energy is so sweet. Lipids or triglyceride, fatty acid chains in your seat. Proteins are amino acids. Peptides make you buff. Nucleic acids, nucleotides keep all your genetic stuff. Good job. Good job. Be prepared to sing it again.
All right, now.
Not today, but again. All right. Let's go over this sheet to make sure you have what you need. And then we're going to go over the questions. And then you will do your exit ticket. No, you won't. Make sure you guys pay attention to when it says you can use your note, when it says you can't use your notes. Because if it doesn't say you can't use your notes, then you can. So take advantage of the opportunity if you need to. All right. So what are the elements found in a protein?
Carbon, hydrogen, oxygen, and nitrogen.
Carbon, hydrogen, oxygen, and nitrogen. So Chun. What's the monomer? Proteins are
Amino acids.
amino acids. See how that kicked it in right there. Amino acids. All right. What's the polymer called? Polypeptide. Cool beans. And the main functions. I told you to write 3 things. What did you write? Structure, catalyst, and immunity. Now that's not all of them. Those are 3 I want you guys to know.
You said what again?
Structure, catalyst, and immunity.
Oh, yeah.
All right. Which picture, guys?
Delta.
Right, it's on the one left. Flip that page over.
Bravo.
Flip that worksheet over. There's 2 monomers on the back. There's 2 monomers on the back. Which one of the monomers, top or bottom, which one is a nucleotide? The bottom. So in that box, you need to identify that as a nucleic acid monomer, which is that nucleotide. What are the 3 parts? That first part that you see on the left-hand side, what is that? Phosphate group. All right. What's in the middle? The sugar, cool beans. And what's on the right?
Nitrogen.
The nitrogen base. Y'all killing it. Y'all killing it. Oh, got We got 19 minutes before it's time to go to lunch. Y'all doing great. All right. I know it seems long, but we almost done. The one at the top, what's that one? That is an amino acid. That is an amino acid. So on the left-hand side, what is that representing? The amino group. I heard somebody say it. They said it low, but I heard it. Either they don't hear things. All right. On the right-hand side, not the middle, but on the right-hand side, what is that? If the amino group is on the left, what's on the right? Carboxyl group. That's the carboxyl group. And what's that thing in the middle? What's that R?
RNA.
That's the side chain. Cool beans, y'all. Cool beans. Cool beans. All right, let's go ahead and go over. Get your whiteboards out. Let's go over page 81. I'll put it up here so you can see it. There you go. All right. Question 1. Proteins. Lord, I done dropped my marker and my whiteboard, so I'm just going to have to say my answer. All right.
One second.
You got it?
Oh, the whiteboard.
Oh, yeah. Get your whiteboard. Get your whiteboards out and your markers. All right. Proteins are found in all cell types and tissues. Contrasted to proteins found in skeletal tissue, what is the main function of proteins found in antibodies? Do y'all know what antibodies are part of?
Your muscles.
No, your immune system. Antibodies are part of your immune system. 1, 2, 3. Let me see. Look at all that delta around here. I thought y'all was going to get tripped up on that one. Good job. Delta. All right, 2. The monomers of proteins are called what? The monomers of proteins. The monomers of proteins. 1, 2, 3. Charlie, oh, y'all was ready. Okay. Thought I was tripping you up again. Let me stop trying to trip. Y'all got this. All right, the diagram shows an amino acid. What is the group containing the nitrogen called? Let's see if we remember this one. What's the group containing the nitrogen called? The group containing the nitrogen. So the side that has a nitrogen in it, what's that called? 1, 2, 3. Bravo! It's correct. You killing it, Cam. Cam up here killing y'all. Ain't got one wrong yet. Alright. Alright. 4. Which of the following correctly states the importance of a protein shape? Which of the following correctly states the importance of a protein shape? 1, 2, 3. Let me say it. Alpha. Love the alphas because alpha is correct. I'm glad it was alpha because if it wasn't, I would have had that wrong. A is for alpha, not apple. We alphas at the top. All right. Anyway, last one. Study the following diagram of a chemical reaction and determine what type of bond is formed as a result of that reaction. Let me see. Delta. Those are 2 amino acids. And what type of bond forms between amino acids? A peptide bond. Awesome sauce. Awesome sauce, guys. You guys did amazing. Make sure you can complete that chart on page 82 without using notes. That way you'll know you're ready for your quiz. If you can answer those questions without using your notes, you're ready for your quiz. If it's something you don't know, look back at your notes, but keep practicing it until you can fill out everything on that page without using your notes. All right, go ahead and do your exit ticket in Canvas. And if you still need more assistance, I have put a video in there that you can watch. And this video will be uploaded after 8 o'clock. It's been nice doing science with you guys. Ms. Young out. Yeah. What's up?
So,
Do we watch the video?
I did,
You can. I put the video, every time I record, I put the video in Canvas. I'm listening.
I tried to do my what's it called, before 10 minutes, but
You need more time? Okay, when I get to my seat, I'll add you some more time. Don't worry about it. Let me go turn this video and stuff off first, and then I'll add you some more time.
Huh? Just don't finish.
Huh?
Just don't finish.
You need more time too? Probably so. Have you had you already opened it up? Okay. Don't worry about it. I'll go back. I'll go check the timing on it. It's no problem, guys.